Hi, everyone. Today we'll be presenting our research on the mutational, phylogenic, and structural analysis of a penicillin-binding protein in novel bacteriophage PhiRho. This is our poster that we're actually presenting in person, but this will be split up throughout our virtual presentation.
So quickly, we'd like to do a brief introduction on our project and why it's important. So bacteriophages are viruses that infect bacteria, and currently, specifically in the medicine and industry fields, there's a rising antibiotic resistance that's happening in bacteria. And so with phage's special property, it has proof that they can be applicable in these industries. And with PhiRho being a newly isolated phage, it has many areas that can be discovered that can help further these industries.
For this project, we decided to focus on gene 176 of PhiRho. We found two major functions of this gene. The first one is a minor tail protein, which are proteins at the tip of the phage's tail that recognize the host and help insert the genetic material. And also D-allo-D-allo-carboxypeptidases, which help build the cell wall and are also known as penicillin-binding proteins. Now we found these are actually really highly related, and D-allo-D-allo-carboxypeptidases are a subfamily of minor tail proteins. And we're also interested in this protein because of its antibiotic resistant tendencies.
So to conduct our analysis of this gene, we analyzed three portions of it. We analyzed its structure using programs called PhiRho and PyMol. We analyzed its phylogeny using PhiML, Splitstree, and Mega. And then we conducted our final mutational analysis using Python coding.
Okay, here you can see the PhiRho predicted structure of gene 176. So you can see it has some secondary structure, including alpha helices and beta sheets. And then in that inset in the right, you can see its catalytic pocket where it can bind either penicillin or the cell wall so it can perform its function.
Here is the superimposition of 176 in a template crystalline structure from PhiRho, and that template turned out to be an octameric penicillin binding protein. And using the superimposition, it has a very high RMS or very low RMSD score, which is preferable. And that means that this gene is most likely part of that penicillin binding protein family.
As you can see in the megagraph to the right, there's many phages listed, and that's because in the fam or cluster of phages, there are over 600 members, so we chose a bunch of different clusters to analyze, with the highlighted versions being those of D-allo-D-allo-carboxypeptidase. We just wanted to see if there was any pattern that could help us predict if it was going to be a D-allo-D-allo-carboxypeptidase or a minor tail protein. We found that there really wasn't any pattern, which could either be due to environmental concerns or just a little error in annotation. And because of this is just further evidence that PhiRho gene 176 is definitely a minor tail protein and could be a D-allo-D-allo-carboxypeptidase.
Finally, looking at a mutational analysis using Python and we compared the two DNA sequences to another gene of another phage. I actually found there weren't a lot of mutations, or only 10 total, but we first looked at the ratio of transition to transversion mutations. Now, the base pairs of DNA sequences are made up of 4 different base pairs or letters, if you think of it. Transition mutations are when there's a change in the same family or pair, and the transversion is when they're a switch in it to a different family. Transition mutations made up about 90%, whereas transversion were only 10%. We also looked at the ratio of synonymous to non-synonymous mutations. Synonymous is when is a mutation, but the amino acid doesn't change, whereas the non-synonymous, it does change. And we found that the synonymous mutations were 70% of those mutations, whereas non-synonymous were only 30%. But again, there weren't that many mutations, which means the sequence was pretty much conserved.
Okay, so as a quick summary of our results, again, the protein was structurally similar to many penicillin binding proteins from archaea and bacteria. This gene is present in many phage, about 600. The sequence is conserved, and all of these things together imply that this gene probably plays a very important role in phage survival and fitness because it's evolutionarily conserved. So in the future, bacterial infections will probably be treated with a combination of antibiotics and phage. So if penicillin could bind to penicillin binding proteins and affect the efficacy of phage, that would be a very important caveat to the treatment of bacterial infections. So this could have important implications for infections treatment in the future. And with that,